So my thesis is based on work done in Professor Andrew Henderson's lab in the BU School of Medicine, and it's about how differential T-cell receptor signaling could impact the establishment of HIV latency. So let's get started. All right, so for some background information, I'm sure all of you are familiar with the human immunodeficiency virus, commonly known as HIV. It's a virus that targets the immune system, and more specifically T cells. And so T cells are a specific kind of white blood cell that are responsible for coordinating your immune response against specific pathogens. So if you don't have T cells, then you're going to be susceptible to a lot of infections and different types of illnesses. It's estimated that there have been about 40 million deaths that have been caused by HIV, and currently there are 35 million people who are currently infected and living with the disease, and there's no known cure. So to understand how HIV latency works, you first need to sort of know how viruses and how HIV works in general. So this is a diagram of an HIV virion. So all viruses are going to have some sort of genome stored as DNA or as RNA, and that will be inside a shell of proteins called a capsid. And for some viruses, this capsid is inside an envelope of lipids. And as you can see with HIV, there are also special proteins that are sort of on the surface of the envelope. And these proteins help the virion latch on to receptors on T cells. And this is what allows HIV to infect these T cells and just specifically target them. And so HIV is a retrovirus. So basically how its life cycle works is, as you can see, the proteins on the surface sort of help it target the receptors on the surface of the T cell. Then the envelope of the virion fuses with the membrane of the cell, and it sort of spills its contents into the cytoplasm. So next, you might have noticed on the previous slide, there are a whole bunch of dots floating around inside of the virion. So what these are special enzymes that basically assist HIV with various steps of its life cycle. So right here, an enzyme known as reverse transcriptase will take HIV's RNA genome and it turns it into DNA. Another enzyme called integrase will basically take the newly created DNA and it inserts it into the genome of the cell. And so now the cell is basically just trying to go about its life and make RNA and then synthesize proteins from this RNA. And after the viral DNA has been inserted into the cell's genome, it will also inadvertently make RNA and as a result proteins for the virus. And then a various collection of enzymes that the virus carries with it will help the various parts of the virus reassemble and sort of bud out of the cell and go on to infect other T cells. And so basically how we combat this is with a group of drugs called antiretroviral therapies. So what these do is they basically take some step in the life cycle and they block it or they inhibit it in some way. So maybe an antiretroviral therapy will block reverse transcriptase or it'll block integrase. Basically, its job is to prevent HIV from reproducing. And so where this creates problems is that although it can stop HIV from spreading to other cells, it can't actually take out HIV once it's inside a cell and once its DNA has been integrated into that of the host cells. And so this is basically related to the problem of latency. So what latency is when a cell is, the DNA is inserted into the genome of the cell and then the T cell goes into a resting state. So it's not creating RNA and proteins. It's actually basically just paused and it's kind of resting. And so this is a problem because the immune system doesn't pick up on this because the cell is not producing any virus. And the cell's not dying because, again, it's not producing anything. It's just sort of sitting there in a resting state. What happens is that when people take antiretroviral therapy, their viral levels drop severely, but it doesn't eliminate the latently infected cells. what happens is that when you stop taking antiretroviral therapy, these latently infected cells will actually switch on and they'll start producing HIV again, and you can have your viral levels rebound in as little as a few weeks. And this can even be after you've been taking these drugs for weeks or years or even decades. It's estimated that you might need to be on antiretroviral therapy for over 60 years for these cells to actually start to decay and die. And so this might be sort of an odd picture, but I thought it sort of gave a nice visual. So this, I guess, is supposed to represent HIV if there was no latency. And so basically what would happen is it infects these cells, it kills them, and then if there's antiretroviral therapy, or if it runs out of cells in a local area, then it just sort of, it stops spreading, and then you're fine. But what happens is it can either actually infect a cell and kill it, or it infects a cell, but then the cell doesn't do anything, and it just stays in a resting state. And this allows it to go on and infect other cells later, once antiretroviral therapy is stopped. So for our experiments, we wanted to first confirm that a stronger stimulus on a T cell will actually produce a stronger signal within the T cell. And then we wanted to look at basically two different populations of cells. One that's strongly stimulated and one that maybe has signaling that's a bit weaker. And basically we want to look at, is there a different number of cells infected between these two populations? And is one of these populations producing more HIV than the other? And then as a final step, we did also want to look at cells that appeared to not be expressing any HIV. So these cells would either be cells that were completely uninfected or they were latently infected. And we would basically re-stimulate these and see if there was a difference in the amount of HIV produced between the two different kinds of populations. So I'll explain more about this very soon. So these experiments were mostly done with a line of T cells, so we weren't taking actual T cells from a human being for most of these experiments. They were done with this long-lasting line of T cells. I think they originate from a cancer patient. And so to control the amount of signaling, basically, we used our own artificial receptors. They're called chimeric antigen receptors. And these receptors had varying affinities to a specific ligand. And so how this worked is that basically, you can see here's the little ligand that fits into the receptor. So if the receptor has a really strong affinity to the ligand, it's going to produce a rather strong signal. And if it has a weaker affinity, then the signal's not going to be as strong. So that's basically how we had these two populations for most of the experiments of the strong affinity, which we would be stronger signaling, and the weaker affinity, which would have weaker signaling. And so then the next question is probably, how do you actually measure how strong the signal is? So you can see here, this is basically a very complicated diagram of a signaling cascade inside a T cell. And it looks very gross, but basically all we did is choose one of these and basically decide to focus on it. So this little colorful block right here, MEC1-2, gets phosphorylated during the signaling cascade. So if we can detect how much MEC1-2 was phosphorylated, then we can see how strong the signal was. If more of it was phosphorylated, then it would be a stronger signal. And so to actually figure out how much of this phosphorylated MEC1-2 there was, we used a process known as gel electrophoresis. And so basically how this works is that you have the contents of cells from one population in one well and the contents of the cells of another population in a different well. And you basically, all these molecules that were in the cell are going to be coated in a negatively charged detergent. And there's a positive electrode down here. And basically all the proteins are sort of dragged through this gel. And the smaller ones that can slip through the gel easily go to the bottom much quicker than the heavier ones that sort of move more slowly at the top. So if we stop the gel running after a certain amount of time, we should be able to know, like, okay, all the cells that are heavy enough to be, or all the proteins that are heavy enough to be MEC1/2 should be like at a specific level, and then we can look at how many there are at that specific level. And so to actually like measure how much there were, we needed to take the gel, which in this diagram is the blue square right here, and we use this Western blot procedure to pull the proteins off of the gel and onto a little nitrocellulose membrane. And this again was done with electrodes. So there is an anode down here and a cathode up here, and basically the negatively charged proteins were pulled from the gel and onto this membrane. And so afterwards, this membrane was blotted with the milk protein so that it was basically covered in proteins, and so that antibodies wouldn't stick anywhere where we didn't want them to. And so we needed to use a primary antibody that actually would bind to the phospho-MEC1/2, and then a secondary antibody that would actually give us a visible trait that we could detect. And then we'd basically measure the presence of the secondary antibody. And so the next step was to actually infect some of these cells with virus. So we chose 2 different populations, high affinity and low affinity. And the virus that infected these 2 populations had a reporter gene, which basically codes for some sort of visible trait. So that way you know that if like there is high expression of virus, then a lot of this visible trait will be produced, and you can basically, in this case it was an enzyme that produced light, so if you measure the amount of light, then you know how much virus would be produced. And in order to measure like how many cells were actually had viral DNA in them, we used a technique known as PCR, which basically just amplifies the amount of DNA and also produces, in our case it also produced chemiluminescence that we also measured, and so we could basically tell how much DNA there was by how much light was being produced in this process. And so the final step, which I won't talk about as much, but basically we took primary T cells. I should mention that this was also done with primary T cells. It was done with Jurkits, which are the T cell line that I talked about, but it was also done with T cells that came from people. So in this final experiment, we looked at cells that did not express this visible trait that the virus had coded for. In this case, it was green fluorescent protein. And so basically, if cells weren't expressing this trait, that meant that either they were uninfected or they were infected, but they were just latently infected and just not expressing this. And so these cells would be re-stimulated with these really like powerful agents, and we'd basically use PCR again to measure how much viral RNA was produced, so like the expression. And yeah, so what we found, the results of the Western blot were a little weird, but as you can see here, this was the weakest, and this was the strongest. These were maximally stimulated, but it's likely that most of the cells in this just died, which is why there's such like a faint, sort of marking where the MEC1/2 should be. And so in here it actually looks like the strongest is in the mid-range receptors, but this is most likely due to experimental error. There are probably a few things we could do differently, but this wasn't the most important experiment. It was basically just to confirm something that we're already pretty sure about. And so this is sort of the interesting data. So what this shows is that in the cells that weren't stimulated at all, you have this kind of very low expression of virus. In the cells that had a low affinity and were stimulated a little bit, there was some expression of virus. But in the cells that had the strong affinity and were very strongly stimulated, there was a huge increase in the expression of virus. But interestingly enough, if you look at basically how much proviral DNA there was, meaning how many infected cells there were in each population, it's really about the same. So basically what this tells you is that in these cells that weren't stimulated as strongly, there is this whole reservoir of cells that are, you know, sort of sitting there and not doing anything, not producing virus. And so it's possible that this suggests that they have a larger latent reservoir. And so for the final part of the experiment, as I said before, we sort of took the cells that did not express this visible trait, meaning they were either latently infected or they were not infected at all. And we re-stimulated them to see how much virus they produced. And so based on what I just showed you here, we would sort of expect that the cells that, you know, seem to have this larger latent reservoir that wasn't doing anything, we would expect them to produce, you know, have much higher expression. But again, the higher affinity cells actually did have a higher expression. And so I'll talk about what this means in a sec. So yeah, as I said before, there seem to be increased levels of phospho-MEK1/2 in the higher affinity receptors, although the data didn't necessarily show that, but it was something that we, for all intents and purposes, basically knew. And there was increased viral expression for the high affinity receptors in the cells in the regular cells and the cells that were re-stimulated. And so for the Western blot, it's possible that the cells that were very strongly stimulated just kind of died, and that's why we didn't see as much. But again, it wasn't the most important experiment. So what the next experiment suggested is that stronger signaling could actually lead to higher expression of HIV, and maybe a larger latent reservoir in the cells that had the weaker signaling. And but then there was sort of some confusion about this, basically because the cells that were less stimulated also, you know, they'd had lower expression after being re-stimulated. So this could mean that there's even deeper latency, like these cells are still capable of producing HIV, but even after being stimulated so strongly, they still didn't produce anything, meaning that they could be capable of it, but we're just not able to reactivate them. Or maybe these cells are actually harboring just defective virus that has a bunch of holes and gaps in the genome, and that just are unable to basically create more virus. And so there are still definitely a lot of questions about basically how latency works and how it's established. So it would be nice to figure out why it's more difficult to mobilize the virus from this low affinity population. We're still trying to figure out the exact mechanism by which latency is established. So why do some cells just go into this resting state after they're infected? And basically, so the whole point of this experiment is to see, is it possible to control the size of this latent reservoir by interfering with TCR signaling? Because this can basically, you know, help create new drugs and basically help lead to eliminating HIV. Yeah, so I'd like to thank Mr. Garber, my thesis advisor, Matthew Gagney, who is the PhD candidate who I shadowed and basically worked under, and Andy Henderson, the PI of the lab, and the BU School of Medicine.
All right, very nicely done, Miles. Questions for Miles?
Dr. Batoff?
Miles, if you had more time
Yep.
to go back and review some of the pieces of this again, what do you think is the most important piece to possibly tweak a bit? If you're going to build out on where you were, so what was sort of the most important next step of this?
Oh, in the experiments? I'd say, I guess, so probably one of these, but I think that in terms of the next like direct step, it would probably be nice to figure out why it's so hard to get the low affinity population to create virus, and basically figure out what exactly is responsible for that, whether we're just not able to make them produce virus, or whether there really is something wrong with the virus in them, whether it's defective or not. Mr. Garber?
What advice do you have for the next EUA student who might be working in that lab that you wish you had known at the beginning of your summer?
Oh boy. Okay. I guess it's very interesting, but there definitely, you do sort of have to do a lot of like self-studying and trying to learn about this on your own, and but it is pretty cool. It's exciting. I got to like work in a biosafety level 2+ room, which meant there was like actual HIV, so I wasn't really allowed to do anything in that room because I was 17. But it was still pretty cool. And yeah, I guess basically you get out of it what you put in. So if you put in like a lot of work and self-studying, then you'll probably be able to like talk about something pretty cool. Yeah. So, yeah.
Her question was why do you have to take medication for so long?
Yeah. So basically, you have to take this medication to keep HIV from reproducing, and as far as we know right now, you basically should be taking it for as long as you can, because that's what blocks HIV, or that's what blocks these latent T cells from, becoming active again. The reason why you have to take it for so long is just that these resting T cells can just remain in that state, ready to turn on again for just that long, for like that many years. So that's why. Max?
Why T cells would take so long to stay latent for so long? Like why it takes 60 years to...
So it's not that, it's basically that they can be latent for around 60 years. And again, that's not like 100% proved. But basically the point of that is that these cells can stay, in a dormant state with the DNA of HIV in them for very, very long periods of time, just because they're sort of not doing anything. They're kind of hanging out, and they can just persist for a really long period of time. Yes?
So how do you think this kind of research is going to contribute to HIV?
Yeah, so basically, I think what this can lead to is basically possible drugs or possible treatments that can help eliminate the latent reservoir. And if that's eliminated, then, eventually after taking antiretroviral therapy, there won't be any threat of like reemergence for the virus. So yeah, this is basically all directed towards trying to find treatments that will actually get rid of the latent reservoir, because right now that's the biggest barrier to the cure for HIV. Jake?
Can HIV still be transmitted when you're taking that medication?
No, actually, so if you've been taking it for long enough and your viral levels have dropped to a low enough level, it's not transmitted, like sexually or however.
All right, one more round of applause for Miles.